Hello students, today we will learn about photosynthesis. In this part, we will learn about the discovery of photosynthesis, the place where photosynthesis takes place, and the light reaction, and lastly, chemiosmotic hypothesis. So, the first part is discovery of photosynthesis. In this part, we have to mention some of the important name of some discoverer who let us know about the photosynthesis mechanism. So the first name we have to take here is Joseph Priestley. He is the person who first discovered that plants purify air, or let's say impure air. So he is the one who first discovered that plants play a very important role in purifying the impure air. Second is Jan Eisenhower. He is a predecessor of Joseph Priestley. He discovered that sunlight play a very important role in this purification of impure air. Then comes Julius von Sachs. He is the one who discovered that glucose is the byproduct of photosynthesis. And the last one is T.W. Engelmann. He is the one who discovered the action spectrum of photosynthesis. So now the next part is, where does photosynthesis takes place? That is the location of photosynthesis. In green plants, we can see the primary place where photosynthesis takes place is the leaf. But the other parts of the plants also takes place in photosynthesis, where we can find this green pigment called chloroplast. But today we will talk about leaf. So leaf contains a particle or a pigment called chlorophyll that is present in the intracellular organelle called chloroplast. You can say chloroplast is the main seat of photosynthesis. That means in here chloroplast, all the process of photosynthesis takes place. So what is the structure of chloroplast? We have already read about the structure of chloroplast in the previous chapters, but here we will get to know a brief structure of chloroplast to go with the other parts of the photosynthesis. So in chloroplast, it is composed of two main components, that is grana and stroma. Now what is grana and what is stroma? Chloroplast is a double membranous organelle within which this grana and stroma resides. Inside of chloroplast, grana is the membranous structure arranged in stacks, and stroma is the liquid substrate on which the grana or the stacks of these grana resides. Grana, a number of thylakoids made up a single grana, and the stroma is the liquid part. So grana is the site of the light reaction. That means grana is the part where the light from the sun is trapped, and stroma is the site for CO2 fixation. That means in stroma, the carbon dioxide gets fixed into carbohydrates or glucose. So from here we can conclude that chloroplast is the seat of photosynthesis. In chloroplast, grana is the is the place where light absorption takes place, and stroma is the part where CO2 fixation takes place. So from chloroplast to to the next part we can go, that is in grana. What makes it possible? What makes it possible to trap the sunlight? What are the mechanism, or what are the different components of grana that makes it possible to trap the sunlight directly? So the answer is LHC, or light harvesting complex and reaction center. Now you will say LHC, light harvesting complex and reaction center. What are these? Basically, these are complexes made up of different kinds of chlorophyll pigments. Now what is a chlorophyll pigment? Chlorophyll pigment is a chemical compound that helps the plants to absorb sunlight. That means that sunlight is trapped by these chlorophyll molecules, but not all the whole sunlight. There is a particular wavelength of light that plant chlorophyll absorbs. What is that particular wavelength of light? That is the red and blue wavelength of light. That is the wavelength of light where plants perform their most photosynthetic activity. So chlorophyll, there are different kinds of chlorophyll, but to perform photosynthesis, there is these two chlorophyll A and chlorophyll B. These two chlorophyll plays the main role in the process of photosynthesis. Other than these pigments, like chlorophyll green pigment, there are other pigments also, but their colors are different. Those pigments are called xanthophyll and carotenoid. Xanthophyll, carotenoid and chlorophyll B. These three pigments forms the accessory pigments because they help chlorophyll A to perform the photosynthesis. So that is why chlorophyll B, xanthophyll and carotenoids are called accessory pigments. And here chlorophyll A plays the most important role in photosynthesis process. That is, the chlorophyll A forms the reaction center. So chlorophyll A is the reaction center, chlorophyll B, xanthophyll and carotenoid pigments, these form the accessory pigments or the light harvesting complex. So now we know what is light harvesting complex and what is the reaction center. Light harvesting complex comprises of chlorophyll B, xanthophyll and carotenoid. Reaction center comprises of chlorophyll A. Both of these complexes, light harvesting complex and reaction center, both of these are present in the membrane of thylakoid. It is embedded in the membrane of thylakoid. So now the next part is light reaction. I already told you that there are two parts of photosynthesis. That is the light reaction. That means this reaction is dependent directly on light. It cannot happen without the sunlight. And another part of the photosynthesis process is the dark reaction. Don't go with the name dark reaction, because dark reaction doesn't mean that it happens during night time. Dark reaction here means that for the dark reaction to happen, it is required that the products of the light reaction must be there in the cell. So the dark reaction is indirectly dependent on the light reaction. Because without the products of the light reaction, the dark reaction cannot go further or cannot start. So here, today we will learn about light reaction. What is light reaction? What are the processes involved in light reaction? So to start with, we have already discussed about LHC and reaction center. So, light harvesting complex and reaction center plays a very important role in the starting of light reaction. When the sunlight falls upon the leaves, here in leaves there are mesophyll cells, and within this mesophyll cell the chloroplasts are present. And as we have already discussed, within this chloroplast there resides light harvesting complex and reaction center in the thylakoid membrane. So what happens when the sunlight falls upon the green leaf? The light harvesting complex absorbs the sunlight and gets excited. The light harvesting complex absorbs the sunlight and it gets excited. It gets excited means that the electron present in its nucleus gets excited. And as we know that the excited electron will pass or will transfer its energy to the next electron acceptor. So here the light harvesting complex gets excited. And we know that reaction center is composed of chlorophyll A. But in plants, there are two types of chlorophyll A. That means there are two types of chlorophyll A that absorb different wavelength of light. One chlorophyll A absorbs 700 nanometer wavelength of light and another one absorbs 680 nanometer wavelength of light. So what is the meaning of 700 nanometer and 680 nanometer here? That means that chlorophyll A can absorb light from the spectrum where 700 nanometer wavelength of light is present. So chlorophyll A absorbs 700 nanometer light and it becomes photosystem 700 or PS1, and chlorophyll A 680 nanometer absorbs 680 nanometer wavelength of light and becomes photosystem 680 or photosystem 2. So there are two kinds of reaction center, photosystem 1 and photosystem 2. Both are present or both are embedded in the thylakoid membrane. So with LHC, light harvesting complex, this is connected with both the PS1 and PS2. That means both the photosystem 1 and photosystem 2 includes light harvesting system of their own. So in the thylakoid membrane, both this PS1 and PS2 is situated. Now what happens? To start with, PS2 680. This is 680 nanometer. This photosystem this photo system gets excited by sunlight. Okay. So this gets excited by the sunlight, or we can say photons. So after getting excited by the photons, PS2, this is the reaction center we have already talked about. So the electron of this reaction center gets excited and jumps at a higher orbital. So this is the excited reaction center right now. Okay. After getting activated by the sun's photon. After getting activated by the sunlight or the photons, this PS2 reaction center transfers its electron to a primary acceptor. After transferring its electron to primary acceptor, this primary acceptor transfers the electrons to a electron transport chain of cytochromes. Electron transport chain of cytochromes. Now, this electron transport chain of cytochromes transfer their electron to PS1 700 nanometer, which is oxidized. Now you will ask me how the PS1 is getting oxidized, because PS1's action spectrum, or we can say activation spectrum, is 700 nanometer. So how can it be reduced? It is oxidized, and how can it be reduced by electron transport chain of cytochrome, which is the electron is coming from the PS2. How it is in oxidized form? So now we will know about the how the PS1 is getting oxidized. When simultaneously PS2 is getting activated or getting excited by sunlight, particularly with the wavelength of light of 680 nanometer. At the same time, simultaneously, this P700 or PS1 is getting activated by sunlight or the 700 nanometer wavelength of light. After getting excited, the same thing happens. It emits an electron. It is now excited, and its electron gets emitted, and this electron goes to reduce NADP, a chemical molecule that is highly energetic. The electron from the PS1 reduces NADP to become NADPH plus H plus. So, while photosystem 1 releases its electron and reduces NADPH plus H plus, while doing that, PS1 becomes oxidized. This is the moment where simultaneously from the PS2, the electron is transferring through the electron transport chain and getting here so that it can fill this PS1 with an electron, because it is here the electron is absent. or it is already it's been transferred to the reduced NADPH plus H plus. So someone or in some way it has to be filled with an electron. So that PS2 is getting that electron to PS1 or supplying that electron to PS1. So now we get the question in our head that PS1 is getting filled by an electron, so it is again reduced. But PS2, after it is emitting its electron to supply the electron to the PS1, it is also getting oxidized, right? It is also getting oxidized. So we have to fill the electron in PS2 also. So how we are doing that? How we will come to the... So we were talking about splitting of water. We already know that PS2 supplies its electron to PS1, so that PS1 can supply its electron to form NADPH plus H plus. But in this process, PS2 is the one which is not getting any electron, or which is remaining vacant. So to fill that vacant position with an electron, a process called splitting of water occurs. What is splitting of water? Here, splitting of water means there is an enzyme called water splitting complex. This enzyme complex splits water to protons, oxygen, and electrons. So these electrons are transferred to our PS2 reaction center so that it can be filled with electrons, because that was in an oxidized state. So water splitting complex is supplying electrons to the PS2. So this splitting of water is exclusively connected with only PS2 reaction center. Okay, so here the splitting of water happens, and water is split into protons, oxygen, and electrons, and that electrons are supplied to where? PS2 reaction center, right? Now if we draw this whole light reaction, then we can see that this is the PS2 getting excited by sunlight, then the primary acceptor, electron transport chain, here is the PS1 that is getting also excited by sun, so electron transport chain is supplying the electron to PS1, the vacant PS1, and the electron that is excited by the electron that is from the PS1, which is getting charged up by sun. At the same time, PS1 also is getting charged by sun, but with a different level, different wavelength of light. So PS1 is also getting excited and emitting its electron to a higher state, and to fulfill or to fill this vacant position is in PS1 reaction center. The electron from the PS2 reaction center is getting there, and this electron from the PS1 reaction center is reducing NADP to NADPH. NADPH plus H plus, right? And the PS2 is getting also vacant. So to fill the PS2's vacant position, water splitting complex here is splitting water to protons, oxygen and electrons, and these electrons are again filling the vacant compartment of PS2 reaction center, right? So this is, if you see this whole process or this whole light reaction, you can see this is getting like this and this. So if you can see, this is in the shape of an inverted Z, the alphabet Z. That is why this whole light reaction is also called Z steam. And because of this zigzag kind of process, this light reaction is also called non-cyclic reaction. Why non-cyclic? Because the electrons we are getting from here, from the water splitting, then to PS2, then to PS1, to NADPH. The electron is not getting back to the PS2 or to the water splitting complex. The electron is getting away. But there is another kind of light reaction happens in plants. What is that? That is called the cyclic reaction. We will talk about cyclic reaction also. In cyclic reaction, PS2 is absent. Okay. So only PS1 is there. And where it is taking place? It is taking place in the stroma lamellae. Now you will ask me, where is the stroma lamellae? You have already seen the ultrastructure of chloroplast. In chloroplast, there are stacks of thylakoids, each representing a grana. And these thylakoids are connected with this juncture that is called the stroma lamellae. Stroma lamellae. So here, the only PS1 exists. No PS2 reaction center, only PS1. So here what happens in PS1, the PS1 reaction center gets excited by 700 nanometer of wavelength of light. Then the electron is passed on, instead of reducing NADP, it is passed on to electron transport chain. And that electron transport chain is producing ATP. I will show you the schematic representation of that. What is happening? This is the PS1. It is getting excited by sunlight. Okay. Then the PS1 rises up to its higher orbital, getting excited. So then this electron is transferred to electron transport chain. While it transfers its electron to electron transport chain, from ADP and inorganic phosphate, ATP is produced. Okay, ATP is produced, and the electron is getting back to PS2 reaction center. So that is why this is called cyclic reaction, a cyclic light reaction, where the electron is not producing NADP. It is getting back to the PS1 reaction center. That is why it is called cyclic reaction, a cyclic light reaction. We can see both of this kind of light reaction in plants. Okay. Now, the next part we will learn about chemiosmotic theory. Now, what is chemiosmotic theory? Chemiosmotic theory. In the past few lectures, we have learned that how NADPH is formed. NADPH is a form of NADPH is a chemical that provides us with high energy. Like in the plant, that provides it with high energy, that performs different kinds of chemical reaction, and it helps in the production of carbohydrate or glucose. But not only NADPH is there, we also know that ATP, adenosine triphosphate, that also plays an important role in production of carbohydrates. Chemiosmotic theory is about that production of ATP, how ATP is produced. The production of ATP is called photophosphorylation. Phosphorylation. In photophosphorylation, what happens is, the first point we have to remember, that for splitting of water, we have seen that from 2 molecules of water, 4 protons, 1 molecule of O2 and 4 electrons are generated, right? Now we have to know that where this water splitting complex is situated. Water splitting complex is situated in the inner membrane of thylakoid. Inner membrane of thylakoid. So we know that we know the structure of thylakoid, that it is made up of a double membrane structure. The outer membrane, there is an outer membrane. Then after that, there is an inner membrane. Within that, there is an intermembrane space. And after the inner membrane, there is a lumen. Okay. So the water splitting complex is situated in the inner membrane. So when a water splitting complex splits water, the protons, oxygen and electrons are getting generated in the lumen of the thylakoid. So this oxygen is getting passed or getting diffused from the thylakoid lumen to the stroma, and this proton is getting accumulated in the lumen. Okay, we will know how these protons helps in the generation of ATP. But first talk about this accumulation of protons in the lumen. Okay, so this is the first way in by which protons are accumulated in the lumen. Then second is when you have to understand, from photosystem 2 from the photosystem 2, electron is getting transferred to photosystem 1, right? Then photosystem 1, we all know that this thing is happening. But while the electron is getting transferred from photosystem 2 to photosystem 1, there is a primary acceptor that is accepting the electron. And then the primary acceptor is transferring the electron to electron transport chain. Here, the components of the electron transport chain, cytochrome is not an electron acceptor. It is an hydrogen. It is an hydrogen carrier. That means that it pumps proton in the thylakoid lumen. As we know that PS2 and PS1 are embedded in the thylakoid membrane. So when PS2 is transferring its electron to PS1, in between, the electron transport chain or the cytochrome complex is pumping proton from the stroma to the lumen. Pumping proton from the stroma to the lumen. Okay? So this is another way by which the lumen of the thylakoid is getting accumulated by protons. Then these are the two ways by which lumen is getting highly accumulated with protons, and the stroma is getting what? Deficient of protons. So we can see if this is the thylakoid, membrane, and this is the stroma, and this is the lumen. Then more protons are getting accumulated here. That means these are more here, and the number of or the gradient of the number of proton is getting decreased in stroma. That means higher proton accumulation here and lower proton is here. So there is generation of a proton gradient, high to low, right? A proton gradient is generated. So how this proton gradient will help us to generate ATP next? So this high proton accumulation in the lumen makes it happen because the protons can be channelized. This gradient can be broken by only the complex called ATPase complex. And now what is an ATPase complex? These can be found in the thylakoid membrane along with PS1 and PS2 and the water splitting complex. ATPase complex consists of two parts, F0 and F1. This F0 part is embedded in the thylakoid membrane, and the F1 part protrudes out in the stroma, part. So the F1 is in the thylakoid membrane, embedded in the thylakoid membrane, and F1 protrudes out in the stroma part. So this protons getting accumulated here in the lumen of the thylakoid can get out from the lumen only through this complex. So when these protons are getting out of this complex, when the protons are getting channelized through this complex, this whole complex, especially this F1 complex, is getting transformed and getting the energy to produce from ADP plus inorganic phosphate to ATP. So now we know that this proton gradient that is happening across the membrane of the thylakoid membrane, that is helping in the generation of ATP in the cell. So this is the chemiosmotic theory.